Okay, so thank you so much for the invitation and introduction. It's my great honor to be here, especially with all the young people, audience, and also the seminar, which are also hosted by Rising Star, who Simon gonna be also my speaker tomorrow. So I'm really delighted. So my name is Tae Seok Moon. I'm currently at J. Craig Venture Institute, and I also hosting a seminar, and that's gonna be tomorrow and every Thursday. So today I'm gonna talk about some sensor applications for the practical goal, and then I'm basically kind of giving some examples. My lab and my collaborator lab basically have been doing for last more than 10 years. So first thing, I mean, I'm shamelessly advertising some, postdoc position at Kraft Center Institute. One thing I wanted to brag, I believe a lot of people right now suffering from cold weather. We are in San Diego, a beautiful temperature and also beautiful view. That's actually the Pacific Ocean. We are overlooking at this moment, and then the weather is almost always sunny. This is my conflict of interest slide. I studied Moonshot Bio after I got Langer Prize. If you have any new idea for the new company, I'm highly recommend just applying it. I believe the deadline coming soon this year, and you can kind of suggest and propose any crazy idea, and then they give up to $100,000. And then, if you don't know, Bob Langer is, I believe, the most famous engineer in history, founding more than 100 companies, including Moderna. And when he was young, also struggled to get the tenure position at MIT. And then he, overcame all the difficulties and then now become the world of the most famous, engineer with a lot of contribution in chemical engineering and also bioengineering. So let me start with my view of metabolic engineering. I was trained as metabolic engineer. My advisor is Christopher Preder. When I studied my PhD after many years of industry experience, I kind of got to know about metabolic engineering, and then people talking about metabolic engineering as a field that basically talking about one feedstock and using typically one microbe and typically in one bioreactor making one compound. That was the definition of metabolic engineering for, I would say, more than 35 years. And I just wondered at the time, why are we talking about this boring one combination rather than thinking bigger? So I kind of wanted to say something at the time, but I was just starting graduate student. I don't want to make some people, angry at me. So I kind of wait until I think 2021. I put one opinion article. I call it SynMade, Synthetic Microbiota Across Diverse Ecosystem. And in that article, I basically, present, it's not necessarily new idea. A lot of people thinking about this one, but I kind of formulate in a sense in the context of the synthetic biology. And then I use the soil as an example, but you can think about water or even entire planet. And now I see the entire planet as huge bioreactor. In that bioreactor, you can have, photosynthetic bacteria that capture carbon dioxide. In that way, potentially harness the bacteria and also, reduce uh, carbon emission or, capture carbon. In that way, potentially reduce greenhouse gas. In that way, climate change can be solved. If you're thinking about, nitrogen fixing bacteria, like, my colleagues from Pivot Bio already done, you can make crop yield increase even in developing country like Africa, and they now adopted this nitrogen fixing bacteria, even GMO, and then put it into the soil, and then they increase the crop yield tremendously. And this is a very important example because, as you can imagine, the world, there are a lot of, problem, including, I call it food inequality. But using this type of the nitrogen fixing bacteria, we could solve the problem like, food yield, increase, and also at the same time, food inequality. And then, I'm not going to talk about this on today, but we are developing plastic eating bacteria, and then plastic eating bacteria potentially release into the world, and then basically, clean up our environment, including microplastic. And then if you have some so-called kill switch, this bacteria might die. And in that way, you don't need to worry about GMO release into the environment, etc. So this is my view, and I'm gonna implement some technology to achieve this goal probably next 20 years of my life. And I hope I want this kind of view kind of adopted by other people, and then the next generation of researcher kind of implement what I thinking about. In that way, this kind of new field or microbiome field kind of continue to prosper. In that way, we can achieve a lot of thing to solve global problems. So the first thing is, I mean, we need to have some tool. So let me explain the first thing first regarding the genomics. If you're kind of working on genomics for a while, we had the tools like knockout tool. There are lots of knockout tool, but knockout tool basically telling you in the single cell, you knock out one gene and then you got to know the function of that gene. We kind of got, those kind of information in E. coli. That means we know almost all the genes function in E. coli and some other model bacteria. So that's how genomics advanced. But do I have such tool in microbiome engineering? Because we are talking about billion different bacteria, and then I'm talking about knocking out only one species or one strain at a time, or combination of two bacteria or two species knocking out. In that way, we could potentially know the function or role of this bacteria or bacterium or even strain. But we didn't have such tool until recently, and multiple groups actually developed such tool, including the Jennifer Doudna's group and also you know Harris Wang's group and then my group recently developed similar tool that basically allow us to engineer microbiome at the species level or strain level. We are basically presenting the strain level resolution engineering tool and by using CRISPR-Cas9. And let me explain this tool, in a bit slow. So, CRISPR is basically molecular seizure that cut the DNA, but in bacteria, bacteria do not have any non-homologous end joining mechanism. That means once CRISPR-Cas9 cut the chromosome, bacteria essentially die. And we're basically using that, idea to basically knock out one strain at a time. The problem is the DNA delivery. And then a lot of people, using, bacterial phage to deliver the DNA because that has at least the species level, specificity. But we want to have the strain level specificity because the phage does not give, strain level specificity. So to achieve that goal, we basically start to use the liposome. That's basically lipid bilayer, and then that encapsulate the DNA, coding the Cas9 and guide RNA. And then this, lipid bilayer or liposome basically non-specifically fuse with any bacterium in the microbiota and then deliver the DNA. And then, of course, the specificity coming from the Cas9 guide RNA. And then that basically means, bacteria only with the specific, design one basically die. So that's basically simple idea. Of course, there are lots of problems, including CRISPR should work in that bacteria, but most of the bacteria, luckily, adopt Cas9 system, and then that means it's matter of optimization, and then efficiency is not high enough, but we start to improve that efficiency. But last problem I didn't talk about is basically how to make sure this guide RNA only specifically targeting one bacterium out of billion different bacteria in the system. And this is the in fact, the simple calculation, basically, as long as you have the sequence information in the system, now we have the tremendous advancement in sequencing, metagenome sequencing. That means we will have those kind of information in the future. That means we probably know all the sequence information in the community. As long as we have that kind of information, we just scan all the sequence information and then we can find very unique sequence only targeting one bacterium in the system. So we apply the same concept with the 4 different, basically the strain. The first thing is basically very similar bacteria system, that E. coli. Sometimes the sequence homology is almost 99% identical, but we basically demonstrate only one guy, in this case, Nisser, basically die, but other bacteria basically survive. We also tested this concept using mouse fecal matter. I'm sorry, I'm talking about, fecal matter just before lunch or after lunch or even before morning or whatever, depending on your time zone. But we also demonstrated only one bacterium die in the mouse fecal matter. This is very targeted level. And then we can use the same technique by adding homologous DNA. And then that means you can easily make the homologous recombination. And then basically using Cas9 as a count selection. But if you have also antibiotic resistant gene in that DNA, you can now select using antibiotic resistance marker. That means you can basically isolate one bacterium out of the billion different bacteria. This is gonna be speed up the isolation process tremendously, because previously to isolate one bacterium that takes sometimes 5 years, including all the characterization. That means one PhD, basically one isolation of bacteria and characterization. But in theory, we can do that within several months if you know the sequence information of your bacteria. So this is the basically the tool we want to utilize, and then I call it the microbiome engineering tools. But more toward, the theme of this, seminar, about sensor, we also need to have some knock-in tool. So we are talking about knockout tool, but we also need to have the knock-in tool. That means in the single bacterium, I'm talking about overexpression tool. But in that case, we are basically using probiotic as a delivery vehicle rather than, health improvement, the tool. So probiotic we use because probiotic is kind of uh, you know, beneficial, but at the same time very safe uh, in the human, application in the future. But we want to have this, probiotic, with the sensor and also actuator. So that means, if you have some disease detected by this, probiotic, that means that this probiotic should have some engineered sensor and then basically generating some color for the diagnostic application. Pam Silver in this, seminar series basically pioneer the diagnostic, probiotic application with the Nature Biotech paper in the past. But this is kind of some of idea, we want to implement. But if you have some therapeutic output, like, curing some disease, we can even go one step further, not just the diagnostic, but basically also kind of potentially sense disease and cure disease, etc. So that is the knock-in tool for the microbiome, because we are talking about gut microbiota or soil microbiota. In that way, you can also, let's say, soil bacteria, you can detect some pathogen using some sensor and then release a pathogen killing molecule. In that way, you can cure, soil environment. I'm talking about some pathogenic, bacteria killing. So to do so, we actually start to develop many sensors, including one of the neurochemical, sensors. Basically, we realized many years ago some neurochemical even found in the gut. Especially one example I always give is serotonin. And serotonin, as you can imagine, uh, actually produce more than 90% in the gut environment, or gut, cell, and then only less than 10%, produced by the brain. And at the same time, I realized serotonin, you know, production enzyme or degradation enzyme also found in the gut. So we wanted to kind of sense serotonin and then make the serotonin or reduce serotonin. That way, serotonin level is kind of maintained in a certain level. In that way, we could kind of make people happy because serotonin kind of control the happiness and then making the serotonin level, right, amount. Of course, this is a very challenging, problem to solve, but the basic idea is we want to kind of, control dynamically in the gut, by detecting and controlling the serotonin level or some other molecule that related to the disease. So, but the problem is this is the one of the, pathway for the all the, neurochemical production. The problem is those compounds structurally very similar. Like one oxygen is basically take off from the molecule, or amine group just gone, carboxylic group just gone. This is so each step, and then the molecule is very similar. That means the making or developing specific sensor is almost the nightmare. Is very challenging to achieve. To make the long story short, I realized I already spent a lot of time. That this is the one of the example of the what we did. We basically use AlphaFold, Rosetta, and some computational tool to design the protein binding site and protein structure. And after many years of struggle, we basically able to produce very specific sensor. I believe more than 10 different chemical sensor. And one example I'm showing here in the right bottom corner, that is basically tryptamine sensor, only, respond to tryptamine, but not respond to other molecule. And then we also develop lots of other sensors, including PEA sensor, etc., as you can see from this heat map. And probably a couple of last point I want to make. So far, I kind of making so-called, GMO, I mean, the genetically modified organism. And I have been doing more than 10 years. And then early more than 10 years, I met one person, not doing science, but ask me question like, okay, you making a lot of GMOs. How can you ensure the GMO in my gut not becoming dinosaur? At the time, I become speechless because this guy basically thinking about me making dinosaur out of bacteria, and then he's worried about that happening. And first thing, how creative this guy is, but at the same time, the general public, understands science in a very interesting way because a lot of media, movies, etc., kind of influence our general public. So I realized my job as a scientist should make sure such concern basically, should be addressed. And then I start to work on the biocontainment more than 10 years ago. And again, the Pam Silver pioneer, developing some kill switch regarding those kind of application. And then I'm not gonna go over very detail, but we still use the same concept, using Cas9 as a killing mechanism, but now in the single cell level. That means the GMO basically having uh, this kill switch. And then, but now we have some trigger, including some chemical, in this case, anhydrotetracycline, because this molecule, is not harmful in the mouse model, demonstrated by multiple, lab, including Pam Silver's lab. And then we decide to use as a demonstration. And then now I have the solution or answer to the guy who asked me about dinosaur. And my answer to him is, if I could make my bacteria becoming dinosaur, I'm gonna become the billionaire tomorrow because I'm making the dinosaur out of the bacteria, or at least I'm gonna get the Nobel Prize tomorrow because this is a breakthrough science, basically. But I still need to solve the problem of biocontainment. So my idea is basically ATC trigger Cas9 expression. That means whenever I'm adding the ATC in the human body or mouse model, and the bacteria basically die in response to the ATC without touching other bacteria, beneficial bacteria, because using antibiotic also achieve the same goal, but killing all the bacteria, and then that result in a lot of other problem, including bowel disease, etc. So that is basically the idea. And then to make the long story short, because it took us more than 7 years because we got a lot of people scooping us, including Jim Collin, my own advisor, Chris Boyd, and also, of course, the Pam Silver. And at the end of the day, we, made the probably best kill switch ever developed. And this is a mouse data. Basically, we are, seeing the control strain not die. Uh, Basically, kill switch strain basically die completely after 2 days in mouse model. And this is basically when we add the ATC or bacteria before becoming the dinosaur, and we achieve that goal. But this is not the real biocontainment because you don't want to spray the ATC after you basically release your fecal matter and then follow all the way to the sewer system. You don't want to spray the ATC to just kill the GMO. So we decided to develop the temperature sensor, and then the temperature sensor basically trigger at the lower temperature, around the 30 degree. As you can imagine, body temperature is the 37. Even in the hot summer, in Africa, outside temperature at night, and this one day probably become the 30 degree. And if the 30 degree achieved, basically the bacteria basically die, and we demonstrate the same thing in the mouse model. And one new piece of the data we wanted to show, we actually combined the kill switch idea with the sensor. In this case, our sensor is the phenylalanine sensor that detect phenylalanine. And then we wanted to decrease the phenylalanine level in response to the high concentration of alanine, because some patient, especially phenylketonuria patient, especially newborn baby, accumulate too much phenylalanine, and sometimes baby die. So this kind of rare disease should be treated currently using diet restriction, but important thing you need to know, phenylalanine is also essential amino acid. We should have certain level of the phenylalanine for growth. So we basically using dynamic control, too high phenylalanine, we basically reduce the phenylalanine in response to the level of the phenylalanine. And then we basically demonstrated that one with the Gautam Dantas at WashU. And then we basically showing phenylalanine level go down with using our sensor response system. And at the same time, kill switch working in vitro. And then of course we use the mouse model, and then phenylalanine still go down, even phenylalanine spike. And then of course our kill switch also working fine in the mouse model as we showed earlier. So this paper under review right now, and I hope this gonna be coming out soon. And last thing I want to tell you is initially talking about this one, we actually started the Symbis seminar series almost 5 years ago, starting with Francis Arnold, and then this became the weekly seminar. If you are interested in giving a talk as a junior speaker, please send me CV or resume to me. And then we have the amazing lineup of the speakers. As I mentioned earlier, Simon gonna give talk as a rising star tomorrow, and Pam Silver already gave a talk as a pioneer. And then we also gonna have the second conference in Barcelona, Spain. We basically inviting all the people in the audience, and also we will have some speaker from this webinar, and including amazing speaker, including Nobel Prize winner, 28 National Academy members, funding agency director, and also many journal editors, including Nature and Cell Journal, and of course, the most importantly, 261 rising stars. Life is tough, and also living as a researcher is even tougher nowadays. If your life is miserable, or you almost want to give up, and I highly recommend you reading this article, one-page article published in Science. I guarantee you will feel better. And also I'm shamelessly advertising my journal, New Biotechnology. This is the European Federation of Biotechnology and Asian Federation of Biotechnology official journal. In fact, factor is not bad, probably higher than some other specialized journal. And this is a European journal originally, but I'm wanting to expand the audience and readers and authors to United States, and I'm happy to take your papers. Consider publication. With that, I want to thank all my students and postdoc and technician in my lab, and also the funding source, and also my two advisors still giving me advice, Chris and Chris. Both are the MIT, department head. Also, thank you for your attention. I'm happy to answer any question you